Hello everyone. Today we are going to discuss about vectors. Introduction. A vector is a DNA molecule that has the ability to replicate autonomously in the host cell and into which DNA fragment to be cloned. It is a biological tool in rDNA technology. It is used for delivery of desired foreign DNA into the host cell. Properties of an ideal vector. It should be replicated autonomously. A vector should be less than 10 kb in size. It should be easily isolated and purified. It should be easily introduced into the host cell. It should have suitable marker genes. Vectors should consider unique target sites and recognition sites for various restriction enzymes. It should have the capability to incorporate either itself or the DNA insert in the genome of the host cell. Types of vectors. Vectors are of many types like cloning vector, expression vector, shuttle vector, cosmates vector, phage-mate vector, and bacteriophage vector. Cloning vectors. Cloning vectors are small pieces of DNA which have the ability and used to introduce foreign gene of interest into the host cell. They can be stably maintained inside the host cell. Cloning vectors are generally used to obtain multiple copies of desired foreign gene. Examples are plasmid, cosmid, and phages, etc. These type of vectors generally contain selectable markers, origin of replication, and restriction site. Expression vector. Expression vector is a type of vector which not only introduces a gene of interest into the host cell, but also aids in the analysis of the foreign gene via relevant protein product expression. It is a type of vector which is used to obtain or analyze the gene product, which may be RNA or protein of the inserted desired gene. Example, only plasmid vector. Expression vector consists of enhancer, promoter region, start-stop codon, transcription initiation, selectable marker, ORI sites, and restriction site. Plasmid vector. A plasmid is naturally occurring extrachromosomal double-stranded circular DNA. It replicates autonomously within bacterial cell. Plasmid carries an origin of replication. Plasmid vectors are simplest cloning vectors. It is most widely used for gene cloning. Characteristics of plasmid vector. It contains an ORI site or origin of replication. It also contains selective markers such as antibiotic resistance, blue-white screening, small in size, which is 1.0 to 250 kb, contains multiple cloning sites, easily isolated from the host cell. Examples of plasmid vectors are PBR322, PUC 18x19, PBR322 vector. It is one of the first vectors to be developed by Bolivar and Rodriguez in 1977. The name PBR322 denotes the following: P is plasmid, B Bolivar, R Rodriguez, 322, differentiated from the other plasmid produced in the same laboratory, which is PBR325 and PBR327, etc. It is 4363 base pair long. It carries two sets of antibiotic resistance genes, that is ampicillin and tetracycline. It contains only single or unique recognition site for 12 different restriction enzymes, PST1, SAC1, and PUV1. This is a diagram of PBR322. PUC 18x19, it is a derivative of PBR322, which is smaller in size, that is equal to 2.7 kb. It is 2686 BP long. The name PUC 18x19 is derived from P is plasmid and UC University of California. Cosmates vector. It is a type of hybrid plasmid. It contains lambda phage cos sequence. Cosmates is equals to cos sites plus plasmid. Genomic size of cosmates is about 30 to 52 kb. If they have suitable origin of replication, then they replicate as plasmid within the host cells. Example, SV40 ORI and COLE1 ORI. It contains selectable markers such as amplicillin resistance gene. Collins and Horn in 1978 was first to describe cosmates. Phage-mate vector, a plasmid vector which contains an origin of replication from a phage. In addition to that of a plasmid, is known as phage-mates. P-blue script SK is a type of phage-mate vector of 2958 BP derived from the PUC19. Phage vector or bacteriophage vector. Bacteriophages are viruses that attack bacteria. The phages are of simple in structure. It contains of DNA molecules having several genes for replication, which is surrounded by capsid. On the basis of structure, bacteriophages are of two types: head and tail phages, example lambda phage; filamentous phage, example M13 phage. This is a diagram of bacteriophage, which consists of capsid head, nucleic acid, collar, whiskers, sheath, base plate, tail fiber, and spikes. Lambda phage vectors. Its genome size is about 48,502 pp. It contains origin of replication, genes for head and tail protein, and enzymes for DNA replication. It has more than one recognition sequence for almost all the restriction enzymes. It should be larger than 38 kb and smaller than 52 kb to packed into phage particles. M13 phage vectors. M13 phage vectors are used to obtain single-stranded copies of cloned DNA. It is 6407 nucleotides long. It is circular and 6.4 kb in size. M13 vector only cause infection in F plus and F cells. It is used to produce several copies of M13 MP series of vectors. Example M13 MP8 and M13 MP9, etc. Shuttle vector. Shuttle vectors are created to replicate in cell of different types of species. They contain two origin of replication in which one is particular for each host species. Also those genes required for their replication and not provided by the host cell. This type of vectors are developed by recombinant techniques. Limitations of vectors. The following are some of the limitations of vectors. Vectors are not very stable due to the changes in metabolic energy, changing pH and temperature in different hosts. The stability of vector depends largely on the type of vectors and host genotypes. Overexpression of a particular type of genes in a host cell is a common problem associated with the use of vectors. The use of a single type of vector might not be sufficient for a particular purpose. The use of multiple vectors is complex and results in difficulties during the process. Even though a large number of studies are done in the field of molecular biology for the production of more efficient vectors, it is time consuming and expensive process. Thank you.